The way we got into this problem was really from what my lab's central interest is, which is trying to understand the natural barriers and mechanisms by which we resist cancer development, tumor suppression. And that led us into the biology of senescence and then into cell therapy. I should say that when I was a graduate student at MIT, I spent a lot of time in the flow cytometry facility with another trainee there then. His name was Michel Saale, and so many years later, we end up working together. So these are my conflicts of interest. Now, I'm going to give you a little bit of background of what we know and sort of what we don't know about this process people call cellular senescence. It was originally defined in the late 50s by a guy named Leonard Hayflick, who was just starting to culture cells in vitro. And what he noticed is that, if you culture cells for a certain period of time, human cells, they eventually stop dividing. They become insensitive to mitogens, they change their morphology, and never divide again. And that goes through sort of this phases. And people wondered whether or not this might be relevant for other things that were of interest, if it wasn't maybe just a cell culture artifact, but something that might be sort of reminiscent or mimicking other processes. One is cancer suppression. Of course, cells that don't divide can't become cancer cells. But there was also interest that maybe this process in some way reflected aging. Again, the fact that cells can only divide a certain amount of time, that could lead to tissue decline and so on. Now, people debated that for many years, but there was some evidence to suggest the anti-cancer mechanism could be important. It was known initially from the work of Jerry Shea and Woody Wright that a DNA tumor virus oncoprotein, SV40T antigen, could extend the lifespan of human cells in culture, and it was known to bind and activate the P53 and RB tumor suppressors. Later, it was shown that replicative senescence, this phenomenon of cells essentially exhausting proliferative capacity in culture, was driven by the fact that human cells don't express telomerase, and eventually the telomeres get short and cause a stress response that leads to this cellular process. We got interested in quite some time ago when we noticed that young cells, if one introduced an activated RAS oncogene, paradoxically they stop dividing. And the mechanism by which they sort of exited the cell cycle and looked very much like cellular senescence. We termed it premature senescence at the time, again consistent with it acting as a barrier that would prevent sort of cells from growing out of control when they've acquired an oncogenic mutation. So in the last years, we've learned a lot about the process, at least this fibroblast process that occurs in culture, and the mechanism by which it carries out. And I think they can be divided into sort of two components. One is this very stable cell cycle arrest. Cells that undergo senescence, I said, don't respond to mitogens. They remodel their chromatin to become where it becomes more sort of repressive at E2F target genes. We think that's part of the mechanism. But what's garnered more attention more recently is the fact that senescent cells upregulate genes that encode factors that are secreted and can modulate the tissue environment. And this was coined by Judy Campisi as the senescence-associated secretory phenotype. It's a gene activation program, involves chromatin remodeling, and a very prominent effect of cellular senescence. Now, again, I think the field of senescence is one that suffers a little bit from murky definitions about what the process really is. But as I said, we think that it's important in pre-malignant settings, preventing the emergence of cancer cells. We can identify senescent cells here, and there's data to suggest that, in fact, it can also contribute to the anti-cancer effects of cytotoxic agents, so these tumor suppressive type programs. But these states are very heterogeneous, and they're not particularly well understood, and so there's still a lot of effort to try to characterize what people mean when they call a cell senescent. Now, one process we got interested in, we noticed now some years ago, is that senescent cells can, through the secretory program that it has, become visible to the immune system. And here's just a co-culture experiment where we have in green fibroblasts that we can either proliferate or trigger to senesce, and we co-culture them with natural killer cells. And what you can see if you look at the middle panel is these red cells will go and attack the senescent cells. And if we genetically suppress the SASP, that's less effective. So senescent cells can be recognized by the immune system, but which cells and how and the way it actually works is very context specific. T cells, NK cells, macrophages, depending on the context. And the rules still are not well understood. We can even see if we can trigger senescence in tumors. You can do this with cytotoxic agents. We can do it genetically, shown here in a model of liver cancer where we can induce p53 genetically. And what you can see in mice, wild-type mice, the tumors will go away. The cells undergo senescence. In vitro, they're just cytostatic. They just sit there. But in vivo, the tumor goes away, and it's an immune-mediated tumor clearance. You can see that in the panel on the right. And here's just a movie of tissue clearing where we're looking at T cells in green. And you can see when we trigger senescence in the tumor cells, we can get massive influx of T cells, cytotoxic T cells that are critical for eliminating the tumor cells. So again, senescence can provoke and modulate the immune systems in a way that leads to immune surveillance, but that doesn't always occur. Many types of tissue damage, fibrosis, other areas where there's chronic injury, can lead to the accumulation of senescent cells in tissues, and it's thought that this can contribute to tissue decline. And in fact, senescent cells accumulate in people as we age, and this is actually a study done some years ago by Ned Sharpless, where he was looking at the expression of one senescence marker, the p16 cyclin-dependent kinase inhibitor, in T cells from peripheral blood of people at different age, and you can see that the correlation of p16 and T cells goes up with age. You can see the senescence cells with senescence markers increase in aged people, they increase in aged mice. But the stunning experiment was done by Jan van Doorzen, where he engineered a model where he could eliminate p16 positive cells from tissues of mice genetically. And he did it in aged mice, and you're just looking at on the right and the left. The left is a 18 or 22-month-old mouse. It's starting to look aged. But if you eliminate these p16 positive cells, the mice gets better. And that sort of was a shot heard around the world, suggesting that if you could eliminate senescent cells from tissues, it might have therapeutic benefits. And people have crossed this mouse model into all kinds of other tissue damage, neurodegeneration, fibrotic, so on and so forth, and tend to get similar results. Oops, I'm going the wrong way. Sorry about that. And so now we sort of think of senescence as having sort of two general and almost opposite effects on tissue biology. One are the beneficial effects. It's actually been linked to normal wound healing responses. As I said, anti-cancer effects. But then there's these deleterious effects where senescent cells accumulate following chronic damage or during aging, where they seem to causally be linked to tissue decline. And so again, we have key questions, I think, that are important in the field, areas my lab is actively interested in, is you have cells at rest, they secrete factors that can be essentially beneficial or detrimental to tissue pathology. And so we want to know what are the molecular features of senescence cells and when they are beneficial and when they're not, what are its physiologic roles, and what factors dictate whether or not the senescent cells are surveyed by the immune system. One might imagine that if the senescent cells are eliminated, you get rid of all the inflammation that's associated with them, and that might be beneficial, whereas if for whatever reason the immune system doesn't recognize them, perhaps because it declines with age, it has negative effects on tissue biology. And of course, we want to understand whether or not we could use this. Now, a big portion of my lab is actually trying to use senescence in tumor cells to enhance immune therapy, to take advantage of the fact that in certain circumstances it can be make tumor cells more visible to the immune system. But I'm going to talk about its use as a senolytics in these other settings where one has chronic tissue damage. Now, there are a lot of drugs that have been described to specifically kill senescent cells, and they've been tested in a lot of preclinical models, and some are in clinical trials. And what I would say is that these do have activity, there's no question about that, but the mechanism of action isn't really well understood. And of course, most of them are repurposed cancer drugs, so of course they have toxicities. And so I had a student that came to my lab that also had worked with Michelle, and she had the idea of perhaps using CAR T cells to eliminate senescent cells as a senolytic. The idea being that Immune cells naturally do it when it works, and maybe harnessing the immune cells when it fails might be a good way to go about it. And so she looked at, for markers that were upregulated on senescent cells, a whole different type of ways we could trigger senescence in the laboratory, and one molecule she found is a molecule called the urokinase plasminogen activator receptor, or UPAR. UPAR is involved in wound healing responses, involved in blood clotting or resolving blood clots in wounds. And interesting, there's a secreted factor, a cleaved form of UPAR that accumulates in blood, and you can actually use it as a clinical test of, say, how advanced chronic kidney disease is. So, like any potential CAR T target, it's not highly expressed on normal tissues. Just showing you the tissue expression. This is human protein data for a number of targets of CAR T cells in development. UPAR, you can see, is not broadly expressed, but it is prominently expressed in a subset of monocytes and macrophages. But it is upregulated in disease, okay? And so here's just staining for UPAR in lung and liver fibrosis. I'll come back to that later. And also cancer. Cancer is actually often have very high levels of UPAR. Again, they're wounds that never heal. So we decided to test whether or not we could develop models of CAR T cells. We developed mouse models of liver fibrosis. This is one with a NASH-associated diet. You can see the accumulation of UPAR positive cells. We can co-stain with a marker of senescence, the beta-galactosidase, and many of those senescent cells are UPAR positive. We've also done a carbon tetrachloride model I'll come back to in a minute. So we engineered, based on an antibody we identified that binds mouse UPAR, SCFEs. We could show, I won't go into this in any detail, that they're specific for UPAR. And then we did an experimental model where we can sort of control induction of senescence, in this case in the liver. And again, it takes advantage of the fact that oncogenic RAS expression, if there isn't a tumor suppressor gene in activation, will actually drive cells into senescent state. So you can do this method, hydrodynamic tail vein injection, where you can trigger senescent cells in the liver. They would be expressing RAS, they're UPAR positive, and if we had the CAR T cells, those cells go away. We see elimination of the senescence marker, SA beta gal, other ones as well. You can see CAR T cells in these lesions. That's the green cells here on the right. We characterize them. They sort of have a high effector memory phenotype, and you know, are all present at the sites of damage. We wanted to go through something that might be more pathologically relevant. These are, again, just some more staining of UPAR in models of fibrosis, liver fibrosis. This one's carbon tetrachloride-induced fibrosis, the bleomycin-induced lung fibrosis model, and so on. And I'm just going to show you the data from the liver fibrosis model, where we engineer mice to have fibrosis. We then treat them with the CAR T cells and then look at what happens. And very much like we heard in the last talk in the cardiac situation, we can see that these CAR T cells go to the organ where the fibrosis is. In this case is luciferase labeling the CAR T cells. And you can see, just looking at serious red staining, that we're sort of resolving the fibrosis. And this is associated with improvements of liver function. So that suggested that this model could be used, or provide, I guess, proof of principle that you could use CAR T cells perhaps to eliminate senescent cells. It sort of very much parallels what Joel just talked about. And we can talk about at the end to what extent some of these cells are activated fibroblasts. But we did return to the Jan van Deurzen experiment, which is he genetically eliminated senescent cells in age mice, because we were curious to whether or not these sort of senolytic CAR T cells would do the same. I don't think we're trying to develop therapies to treat aging, but aged mice have a lot of things that go wrong, and you can test them all at the same time. This just shows you, and again on the bottom panel, we have a co-immunofluorescence of our target, UPAR, and the senescence marker beta-gal. These is in different tissues in young mice. You don't see it because there's not cells there. But as mice age, you see UPAR positive cells accumulating in tissues. They're often in areas where you also can detect age-related fibrosis. And they co-associate with the senescence-related marker. I should also say that soluble UPAR goes up in mice as they age, and it goes up in people, too, as they age. Now, we've been able to sort out the UPAR positive cells, do single-cell RNA sequencing. I'm not going to show you any of that data, but suffice it to say, we definitely see fibroblasts. We definitely see macrophages that are positive for UPAR. Interestingly, they're always in the sites of tissue damage. They're much more prominent there than any other place. So there's probably some coordinated program going on there. But then depending on the tissue, there's other cell types that also express these senescence markers and co-expressing UPAR. So we certainly don't know what is the disease-driving cell in any one of these tissues. It's something we're actively working on. In any case, we took old mice and gave them the UPAR CAR T cells, about 20 months old, and then just monitored the persistence of these CAR T cells over time. As controls, we use untransduced T cells and then CAR T cells that express the CD19 targeting the human CD19 that we've heard a lot about today. Interestingly, compared to the controls, the UPAR CAR T cells expand, which of course would be consistent with antigen engagement. We see that at 20 days. We see that at 2 months. And while these mice don't live significantly longer, although I have to say our studies are probably underpowered for that, they don't live shorter. And one thing we can measure is inflammatory cytokines in the blood. And you can see on this heat map here, if we just have the control CD19 compared to UPAR CAR T cells, we're reducing the amount of inflammatory cytokines in the blood, which is consistent with these CAR T cells targeting cells that would be secreting such factors. Again, this is just an example looking at 3 tissues where we can see the reduction in the senescent cells following treatment with the UPAR CAR T cells, irrespective of basically the organ. But the main thing we focus on, we put these mice in sort of exercise tests, and they actually showed significantly better sort of ability to run on a treadmill and things like that. But the most striking phenotype that we've gotten so far to date really relates to sort of age-associated metabolic syndrome. And I'm just showing you here results from the glucose tolerance test. We've done many others around this pathology. But just showing you that if you look at baseline glucose levels, if we treated mice with CAR T cells, it's lower. And if we look at the glucose tolerance test, and again, you're looking at the red line here compared to the other 2, there's a much better response in this assay. And this looks exactly like a normal young mouse would look like. Now, we also treated young mice with the UPAR CAR T cells and did the glucose tolerance test, and it didn't get any better. It was already good. But we aged those mice and waited, 18 more months, and then did it again. And again, they only got 1 dose of CAR T cells at 3 months. We analyzed it at 18, and you can see that there is an improvement in the glucose tolerance test. So these are acting in a prophylactic way. And in fact, we see the total levels from young mice of the CAR T cells increase in the peripheral blood as the mice age, again, consistent with the target and antigen sort of accumulating. So very, very interesting. We didn't see any pathology. And again, I am surprised by that because we would have expected, an effect perhaps because of the monocytes that express UPAR, but we really couldn't see anything. All right. We decided, because it's hard to get a lot of old mice, and we would try a sort of surrogate experiment in young mice where we give them high-fat diet. That will give them sort of a metabolic type syndrome, and we can treat them with the UPAR CAR T cells, and they then see an improvement in the glucose tolerance test. And just like we did with aging, we can also treat the mice before we put them on high-fat diet, and then ask what happens in this assay. And again, they show improvement. So the CAR T cells in the system are able to function prophylactically. Now, we're excited by this because, again, we're seeing phenotypes in what is a very complex biology. As I said, I don't think we're going to be taking CAR T cells to treat aging anytime soon, but it does suggest that cell therapies can have beneficial effects on pathologies that are quite complicated and something that we're continuing to pursue. Just going to spend the last couple slides saying, okay, well, how are you going to clinically develop this, right? As I said, people aren't going to do this for aging. The clinical trial will take forever. Even in fibrosis, you know, it's a challenge to think about. But we are plowing ahead. We did develop our own UPAR binders that bind the human UPAR. We actually tried to get them to bind what's called the D2, D3 domain. This is more likely to be retained on the cell surface if there's cleavage of soluble UPAR. And we can show in cytotoxicity assays, we have a whole bunch of binders that are quite good at limiting cells that overexpress UPAR. If we knock out UPAR using CRISPR on the right, you don't see any cytolytic activity. We've also started to characterize in much more detail, and I'm just going to show you snippets of this using spatial imaging now, different types of indications that we might be thinking about. Liver fibrosis, here again, you can see the UPAR positive T, whoops, I guess it's on time, overlapping. We also see high levels of UPAR, again, sometimes in fibroblasts, but not just in fibroblasts. In the lung. But interestingly, and I alluded to this before, UPAR has long been known to be overexpressed in cancer. And there's a lot of cancers that overexpress UPAR, and it tends to be at very high levels. And so it raises the possibility that perhaps a place to start with such CAR T cells might be in cancer patients. We've done a lot of work on mouse models of ovarian cancer, so we started to look at ovarian cancer. I think pancreas cancer actually is probably the highest expression of UPAR, but in any case, a number of patients, samples on the left, and then cell lines on the right, have high levels of cell surface UPAR expression. And we've characterized a lot of human high-grade serous ovarian cancers to look for those where it's uniform, high, and so on. And then we tested in these models where we've either done, we've typically done orthotopic or we've done metastatic models of high-grade serous ovarian cancer in mice and treated with the CAR T cells. And again, you know, these are mouse models, but we see really every single mouse, the tumors get completely blown away and eliminated. You can see the protein, this is just immunohistochemistry. In the orthotopic model, you can see metastases to the lung. They're also eliminated by the UPAR CAR T cells. And we've done sort of a re-challenge experiment where we've looked at, we've had a dual flank situation where we treat with the CAR T cells, the tumors go away. And then in one flank, we inject with cells where we deleted UPAR, and the other flank, we inject the parental cells. And what happens, you can see there's both, the blue line are where we have the parental cells, they express UPAR. Obviously, they aren't forming tumors, but if we've deleted UPAR, the target for the CAR T cells, now they grow. So again, these are persisting at least in the course of this study. Now the last experiment I'll show you is one again that we've done with, all of this work has been with Michel's lab, but his postdoc Sasha Hubner has adapted an assay which have been used by others of reconstituting human immune systems in immunocompromised mice. And again, given the fact that monocytes and macrophages are the main normal cell where we're seeing target, we thought this would be at least a first attempt at a safety type of experiment. And so the idea is we make the humanized immune system. We wanted to give them a tumor so that the CARs would be hyperactivated, so we use the ovarian cancer cells. We also put CD19 on these ovarian cancer cells so we could use the human CD19 CAR T cells. And if we just look at the tumors treated with these, you can see that the CD19 CARs and the different UPAR ones are causing eradication of the tumors, at least most of them completely, but in the case of the blue one, T5, it's only transient. And so at that time, we looked to see what happens in the peripheral blood. And here's just looking at some of the data where you can see in the B cell compartment, the CD19 CAR T cells are eliminating the B cells. That says the experiment worked. But we're really seeing in the monocyte and macrophage population no depletion of these cells. We know there are UPAR positive cells. I actually don't know why they aren't all going away. Maybe they get made faster somehow. Then they're eliminated. But nevertheless, I think this is promising that these CAR T cells might be safe. I think where we're probably going to end up is, of course, all of you know that many cancers arise in fibrotic tissues. And because cancers overexpress UPAR and the surrounding fibrosis has UPAR-positive cells, we think it might make sense to try to go into these patient populations where there would be a strong rationale to try to treat the tumor, but we also might get a signal on the surrounding fibrosis. Work from Elaine Pure has actually shown you can target fibrotic cells in tumor environments, and so I think this is a interesting point. And so I think I've gone on long enough, and just want to finish. But again, we think this and together with, of course, some of the tricks Michelle and all of you can do with CAR T cells in terms of their design might eventually lead to paths forward. So with that, I just want to stop. Thank you, and take any questions.